Chromatography encompasses a group of separation techniques where molecules distribute between two phases, a stationary phase which they tend to stay in and a mobile phase which they favor. In column chromatography, the stationary phase is packed into a tube column and the eluent mobile phase is pumped through it. Sample components travel at different velocities, ultimately being detected and collected at the other end of the column.
Next, I'm going to talk about how paper chromatography works. The first step is capillary action, which is the capability of liquids to flow through narrow spaces against gravity. Capillary action in paper chromatography causes the solvent to migrate up the paper strip due to its attraction to the paper fiber. The solutes in the examined mixture are carried by the solvent as it travels upwards. Adsorption and partitioning are the two major processes driving solute movement. Adsorption happens when the solute molecules stick to the paper's fiber or other surfaces. This process can be affected by the polarity and the size of molecules. More polar or larger molecules tend to interact more strongly with the stationary phase. On the other hand, partitioning is when solid molecules spread between two layers that don't mix. In this case, the paper stationary phase and the solvent mobile phase. How much the solute partitions depends on the factors like its polarity and how much it prefers each phases. As solids move up the paper through capillary action, they undergo varying levels of adsorption and partitioning, leading to their separation based on their characteristic. By examining how far each component travels on chromatogram, scientists can determine properties like retention factor values and identify unknown substances by comparing them to known references.
Chromatography is a biophysical technique that enables the separation, identification, and purification of compounds present in a mixture for qualitative and quantitative analysis. The compounds to be separated are distributed between two phases, one of which is called the stationary phase, while the other is called the mobile phase and moves in a definite direction. Chromatography is of two types, column chromatography and planar chromatography. Paper chromatography is a subtype of planar chromatography that uses a superior quality filter paper, usually the Whatman No. 1 filter paper, as the stationary phase. In paper chromatography, the stationary phase consists of a layer of cellulose highly saturated with water. Water droplets settled in the pores of the paper make up the stationary liquid phase. The mobile phase is usually a polar or a nonpolar solvent or a mixture of both and travels up the stationary phase carrying the samples or analyte along with it. Paper chromatography is therefore also referred to as liquid-liquid chromatography. The choice of solvents to be used in paper chromatography is varied and depends primarily on the substances to be separated. As far as plant pigment chromatography is concerned, one can use a variety of solvents including ethanol, methanol, isopropanol, acetone, and petroleum ether. However, green leaves contain a mixture of pigments with varying solubilities in either polar or nonpolar solvents. Therefore, for optimal separation of the pigment, we'll be using a mixture of both a polar solvent, which is acetone, and a nonpolar solvent, which is petroleum ether. Paper chromatography is subdivided into various types, such as ascending, descending, circular, and two-dimensional paper chromatography. In this video, we'll be employing the ascending type of paper chromatography. For this experiment, we'll need the following: chromatography paper, chromatography jar with lid, petroleum ether and acetone mixture in a 9:1 ratio, threads and tapes, pestle and mortar, sand, some fresh green leaves, funnel, filter paper, small beakers and vials, capillary tubes, scale and pencil, and a pair of scissors. To begin the experiment, take 2 to 4 fresh green leaves. Discard the midrib and grind the rest in a small amount of acetone petroleum ether solvent mixture along with some clean pre-washed fine sand to aid in the grinding process. Add more solvent if required. Filter the extract and collect the supernatant in a small container. Keep this extract aside and proceed with preparing the chromatography paper and chamber. Take a strip of Whatman filter paper pre-cut to approximately 5 by 20 centimeters. Draw a pencil line about 1 centimeter above one end of the paper. Make a V-cut below the pencil line like so. Meanwhile, take the chromatography jar and stretch a thread across the diameter of the mouth of the jar and secure with sellotape. Now, take the chromatography paper and using a fine capillary tube, apply micro droplets of the leaf extract at the center of the pencil line. Allow the spot to air dry and then add a second drop. Allow to air dry again and add a third drop. Repeat the application for 5 to 10 times. Now, make a fold on the other end of the paper. Place the paper on the side of the jar and determine the height of the paper relative to the depth of the jar. Make adjustments in such a way that the pointed tip of the paper lies below the solvent level in the jar, but not the spot. Now, pour the solvent, which is a 9:1 petroleum ether acetone mixture, up to about 2 centimeters deep into the jar. Carefully lower the paper into the jar and use the fold to hang the paper on the thread. Cover the jar and allow the solvent to rise up the paper. As the solvent runs, you will observe that the leaf extract that was previously applied onto the paper starts to travel upwards along with the solvent, and as it travels, gets separated into its constituent pigments. Once the solvent reaches a desired level in the paper that isn't too high up but not too low either, remove the paper from the jar. Immediately mark the solvent front with a pencil and allow the paper to air dry. Mark the farthest distance traveled by each pigment on the paper. This is called the chromatogram. The green leaves used in this experiment were found to have 4 different pigments, namely chlorophyll B, chlorophyll A, xanthophyll, and carotene. Additionally, you might obtain a 5th pigment, namely anthocyanin, if you have used leaves with reddish coloration. Using a scale, measure the distance traveled by each pigment and calculate the retardation factor, or RF, for each pigment using the formula RF equals distance traveled by pigment divided by distance traveled by solvent.
Chromatography is important to industry for a number of reasons. Pharmacists that calculate the concentration of substances in new medicines are one of them. Afterwards, we have the hospital. In hospitals, blood and alcohol levels are detected using chromatography. Furthermore, law enforcement compares samples from suspects and samples taken from crime scenes through chromatography. Environmental agencies also employ chromatography to detect the quantity of contaminants in water supplies. Lastly, chromatography is employed as well in manufacturing facilities to purify the chemicals required to create the product.
To conclude, this experiment on paper chromatography successfully demonstrates the technique efficacy in spreading and identifying different pigments in a mixture. The results show distinct separation of the components such as pigment chlorophyll A, chlorophyll B, xanthophyll, and keratin, then confirming the presence of multiple substances based on their unique RF value. The experiment highlights the principle of solubility and capillary action, which govern the movement of substance on the chromatography paper. The reproducibility of the result underscores the reliability of paper chromatography as a method for qualitative analysis in various fields, including analytical chemistry, medical research, and environmental science. Future experiments could explore the use of different solvents or more complex mixtures to further understand the nuances and application of this versatile analytical technique. Thanks for watching.